We said that viruses can't replicate outside the host cell, and they need a host cell to continue living and a kind of living and to replicate. So how viruses can replicate? Viruses replicate through two ways or two cycles. First one is called lytic cycle. In case of lytic cycle, the replication happens very fast and cause active infection, meaning that the symptoms of the disease can appear on the infected person within few days, ranging from 1 to 4 days from being exposed to the infection. An example of this active infection caused by viruses which enter directly lytic cycle is common cold and influenza. Okay, so let's see how this lytic cycle happens. This is the lytic cycle. In this case, the virus get attached to the host cell, enter by endocytosis, and when it enters the cell, the capsid coat surrounding the virus will get lysed, and the virus will use the components of the host cell itself to replicate the genetic material, which is DNA or RNA, into many copies of it. Then it will also use the host cell components to form capsid coats which will surround the new copies of the genetic material. Then it will get outside the host cell by either exocytosis, where it will take part of the host cell with it, and in this case the body will not identify it as a foreign material because it is surrounded by part of the host cell membrane itself, or by the cell lysis, where it will cause rupture of the host cell membrane to get outside, and from this host cell it will be transferred to other host cell to cause infection. The other way is called lysogenic cycle, and it's different from lytic cycle because it will cause not active infection but latent infection, where the disease can remain dormant for long period of time, for months or even years, and it can appear by certain circumstances like a drop in immunity or by a stressful situation. Then the disease will appear. Example of lysogenic cycle is the herpes simplex. It can remain dormant for many years or many months, and then it will appear when immunity system is dropped, and it will cause cold sores. So this is the lysogenic cycle. The virus in this case will enter the host cell, but instead of replication like the lytic cycle directly, it will combine with the genetic material of the host cell itself. Okay, then it will start replication, but in this case the cell itself will replicate carrying the genetic material of the virus beside the genetic material of the host cell, and in this case the genetic material will be completely changed. Then, like we said, when a drop in immunity occurs, the virus or the genetic material will be activated and it will enter the lytic cycle and the disease will appear. As for retroviruses are the viruses which can carry the RNA only as a genetic material. We have many famous known retroviruses, of course, like COVID-19, it's a retrovirus, or HIV, which is also very famous and very known retrovirus. And in this case, the virus can remain dormant for a long period of time, and it will appear causing a severe drop in immunity. This is the structure of the retrovirus. It has a single strand of RNA, and this is an enzyme called reverse transcriptase, which help the virus to transcribe the RNA into DNA that can combine with the DNA of the host cell inside the nucleus. Okay, let's talk about prions. Prions is a unique case of diseases also, but not the same as viruses. Prions are normal proteins that exist normally inside the cells of humans, but in some cases these prions can have certain mutations that cause serious diseases. Okay, so when we say proteinaceous infectious particle, this refers to prions. Prions are proteinaceous infectious particles. The normal shape of prions is a coil shape. So what is the mutation that can occur in these prions? Mutation occurs in the shape of the prion itself. When a mutation happens, it will cause misfolding of the protein, leading to some shrinkage. Okay, and these are examples of diseases caused by prions. We have mad cow disease in cattle, scrapie in sheep, and Creutzfeldt-Jakob disease in humans. This is the shape of a patient with Creutzfeldt-Jakob disease. You can notice here that there is a space caused by brain shrinkage due to deterioration of these prions. So this is the shape of normal brain, and this is the shape of brain infected by mutation in prions, leading to shrinkage and leaving spaces in the brain.